Good day. My name is Glenn Pierce, and I'm the Vice President of Medical for the World Federation of Hemophilia. It's my pleasure to give you a brief update today about the progress that we've made as of July 2021 in gene therapy for hemophilia. Let's start with some common information. What is a gene and what is a protein, such as factor VIII or factor IX? Well, genes are what make us who we are today. They're the stuff of life. They're what make us into the human beings that we are. We have around 30,000 genes, and these genes are organized on chromosomes. We've got 46 chromosomes in each of the cells of our body, and those cells then are designed to express some of those genes as proteins. In order to provide the function for that cell and to make up the entire function of the body. So within the cell, the genes make proteins, and these proteins, some of them are excreted, and like factor VIII or factor IX, they'll go into the circulation and function, in the case of these two proteins that we're talking about, to clot blood. If there's a malfunction in the gene, if the gene isn't working right, then the protein is not made correctly, and we have an abnormal protein that doesn't work the way it's supposed to. Or it may not even be made if the gene is badly damaged. And in that case, we have hemophilia. So there's different types of gene therapy that is designed to fix a gene or replace a gene. We have gene editing, which has been in the news quite a bit, known as CRISPR-Cas9. And what that does is that precisely replaces a defective amino acid, or in this case, nucleic acid, inside of the gene, and then thereby fixing it and repairing that gene so that it now functions normally. We also have cell therapy. Cell therapy we've heard about in the common news as well. And it induces a working gene by bringing it into the body with a cell. And so that cell then delivers the new gene to the body. There's actually a clinical trial ongoing in hemophilia that is designed to do just that, and to deliver factor VIII into the abdominal cavity through a cell. And then there's traditional gene transfer. It's gene therapy where we know we have a defective gene, we just add a gene to replace the function of the defective gene, but the defective gene remains in the body. And so that's what we'll concentrate on today, and that's the most common form of gene therapy. Well, gene therapy has been around, at least in concept, for 50 years. There have been more than 2,500 gene therapy research projects that have been completed or are ongoing or are in animal testing. And we're continuing this research. 2 of these gene therapies have been approved in the United States. One is for gene transfer therapy for a genetic eye condition. The other one is for a disease, a lethal disease in small children called spinal muscular atrophy. And it's designed to circumvent this gene and allow the patients to normally develop and make their muscles function. So 2 good successes along the way. We have had some gene therapy trials in hemophilia. In 1998, the first trial started for hemophilia B. And then in 2015, we had our first successful gene therapy trial in hemophilia A get started. So how are these new treatments, such as gene therapy, studied? Well, they're studied initially in the laboratory, and that can take several years to even 10 or 15 years sometimes, to develop the questions that need to be asked, work with the animals and the cells in the laboratory to be able to answer those questions, and then look at safety, look at efficacy. And then once all of that has been established, then we move into human studies. With the human studies, the first ones are called phase 1, phase 2 studies, and those last maybe a year, year and a half together. And they look Either it's small studies in healthy people or in individuals that actually have the disease. In the case of gene therapy, we look at individuals who have the disease, such as hemophilia, and do our initial studies in patients. And then assuming those initial studies look good, then larger studies are done. And those studies then are the ones that are submitted for regulatory approval. And then the product can be released and sold for patients in individual countries. After a product is on the market, as you may know from some of the other hemophilia protein products, there's often a number of phase 4 studies, and those are additional studies to understand more about the gene therapy in this case, so that we can do a better job in looking and monitoring for safety as well as for improving efficacy. So how do we actually transfer a gene into a human body? Well, we use viruses. Viruses for really 500 million years have been getting genes, their genes, into the bodies of not humans 500 million years ago, but animals. And so they've gotten very good at it. Animals, on the other hand, have gotten very good at defending against these viruses. And so it's been a struggle over 500 million years. But we're able to take one of these viruses, manipulate it, and use it to deliver our genes of interest. So that's what we do. We take a virus, in this case it's AAV, adeno-associated virus, and we take out its own genes, throw those away, and we put in our gene of interest, which in this case is factor VIII or factor IX. And then that serves as a vehicle to deliver our gene into the human body. There are other viruses that have been used as well, but for hemophilia, most of the work has been done with the AAV virus. So what do? Well, it's shown in this diagram here. The DNA goes, such as factor VIII or factor IX, goes into the virus, the AAV, and then it's just injected intravenously into the body, where much of it goes into the liver. And in the liver, it goes into the hepatocytes, which are the workhorse cells of the liver. And inside those hepatocytes, this virus gets the DNA into the nucleus of the hepatocyte. And in this case, the DNA then goes on to make RNA, protein. The protein gets secreted, and we can actually measure that protein in the circulation when we are successful with the gene therapy. So it's a simple process, but the actual translation of that process into making factor VIII or factor IX that we can measure has been much more complicated. So the studies that we're doing really determine if we can introduce the functional gene into the body of patients who have mutations that are responsible for a whole variety of genetic conditions, including hemophilia. And then we ask, once we get the gene into the body, can we produce protein? Can we detect it in the circulation? And by doing that, then we are able to look over a long term for both the positive as well as any possible negative effects of doing this kind of a therapy. And so for people with hemophilia A or B, we want to see if the body can now make the factor VIII or the factor IX that we've introduced as a gene from the virus. And so once we then do that, then we look to see not only that the factor VIII or factor IX can be made, but that it actually works. It prevents bleeding from occurring. And in that sense, then it lessens the physical situation, the mental and emotional burdens of having to deal with hemophilia every day and night. And it allows us to look at the quality of life, and we hope will change the quality of life if individuals are treated. So a lot of things that go into gene therapy. What I've talked about is really just administering it, but after that, long-term patient monitoring is needed, as well as some pretty intensive short-term monitoring. Not every clinical site can do gene therapy, and so the clinical site needs to be prepped and ready to be able to do it. A lot of conversations need to occur with patients to make sure that we all understand what it is and what it isn't, what it can do, what it might do, what it may not do. and we need to be identified. Are we a good candidate? Do we have the right characteristics to be able to participate in the clinical trial? And so gene therapy risks and benefits really depend upon where one lives. We are very fortunate to live in a very high income country, the United States, like Canada, Western Europe. We have a high income throughout the United States. And therefore, we have medical treatments that are not available in the rest of the world. As you go down into the green areas, they don't have treatment for hemophilia. We run a humanitarian aid program at the World Federation of Hemophilia that supplies product to many patients in the developing world, 20,000 a year actually, but they don't otherwise have full access to treatment the way we do at all. So for them, the idea of a once and done treatment, the risk benefit ratio is very favorable because many of these kids die before they reach adulthood. So that's what happens in a low middle income country or a low income country. And they would be interested in any form of treatment that would be available. As we move up the economic scale and we go into a high middle income country, then more treatment would be desired because most of these patients don't have enough treatment to be able to do prophylaxis to prevent most bleeds from occurring. And then when we look at a high income country, we have great treatments. And so what we're really interested in is a cure. We don't want to have to deal with our constant prophylaxis therapies or the occasional breakthrough bleed. So while everybody wants a cure, some people just need treatments as well. Whereas in the high income countries, a cure is really the next step that's needed. So what is a cure? Well, it means no longer being dependent upon replacement therapy. And ideally, it's converting a severe individual, severe hemophilia patient, to high, mild, or a normal patient even, greater than 30% factor 8 or factor 9. But even converting a severe to 5 or 10% would be a dramatic improvement, and it would result in an incredible increase in life in the low-income countries. So we need to think about things in terms of the risk benefit for individuals that have access to treatment versus no access to treatment. So where are we at? Well, we've got 2 phase 3 trials that have completed and are now in follow-up phase, one for factor 8 and one for factor 9. And I'll briefly mention both of them. The factor 8 trial is with valoctocogene roxaparvovec, and it's using an AAV vector to deliver the factor 8 gene, the B domain deleted factor 8 gene. And what we can see in this trial with 112 patients enrolled is that over the course of a year, the factor 8 level begins to come up, stabilizes at around 17 to 20 weeks, and then remains fairly stable over the course of this year. If we look at the bars here, I'm about a 30% factor 8 range. We can also see that there's wide variability. There are a few patients that are at 0. They've not had a response to the therapy, and it's really not known why. And then other patients have higher than normal levels. The normal levels are 50 to 150%, shown in the shaded bars. And so some patients have gone far above those normal levels. If we go out 2 years in a smaller cohort of patients, we can see that in the second year, there's been about a 50% drop in the amount of factor 8, from 30%, well, down to 20% or so. So maybe about a drop of a third or so. And so these patients are continuing to be watched to determine if the drop continues, because durability is very important. Now, does it work? Yeah, it does work. Looking at the bleeding rate, we go from before therapy, if we just look at the median levels, 2.8, down to 0 for the median level of bleeds after the therapy. And about 80% didn't bleed at all after the therapy. That also means 20% did bleed. And so this isn't perfect. Some patients are still continuing to bleed. If we look at the amount of factor 8 that's utilized, these patients were taking about 130 injections a year. They were all on prophylaxis. And what we can see is that the mean number of injections during that first year dropped to 2. And so while it wasn't perfect, it's a substantial drop. And the number of subjects that were factor VIII infusion free during the course of that year was about 2/3 of the patients, where 1/3 still required some factor treatment. What about the factor IX? Well, that's a tranocogene dezaparvovec. That's also an AAV5 adeno-associated virus, and it's delivering a mutated form of the factor IX gene that has higher activity in it. And so if we look at these patients, they averaged out at around 12 weeks to 4 months at about 37% factor IX activity. And then that persisted at least over 6 months. And by the time we get to a year, there aren't very many patients left. So we don't know how long it will last. But based upon a number of other small factor IX studies, it looks like it lasts for many years. And so we would expect that to be the case here. A number of the patients in both of these trials needed to take steroids because their liver enzymes elevated as a result of getting this therapy. And it was generally well managed, but some patients needed to take the steroids for months at a time, especially in the factor VIII trial. Does the factor IX work? Yes, it does. When we look through 6 months, we see that about 70% of the patients had no bleeding at all. That means 30% did have a little bleeding. So there is some breakthrough here as well. So where are we now? Well, for hemophilia B, from all of the trials, we see that we have stable expression for at least 8 years and counting. We see that this mutated factor IX can get us into the 20, 30, 40% range for factor IX activity and really is enabling for gene therapy. We see stable expression. We have variable responses here too, from 1 to 109%. But we also see that bleeding is controlled in most of the instances. There are still some unresolved safety questions about the liver enzymes and about the potential for long-term cancer, but nothing has been detected so far, and patients are by and large doing very well after therapy. For factor VIII, the situation is essentially similar with a couple of exceptions. Bleeding is controlled in most instances. Most patients do have therapeutic expression at 1 year, but we have a loss of expression that has been seen in an earlier trial over a 4-year period of time, with about 75% of the initial activity lost. We see a lot of variability, more variability than we actually see with factor IX. The liver enzymes are elevated in most of the patients here. And so there are safety issues that still need to be considered as we look at the long-term outcomes in these trials. These trials are heading toward FDA review and presumably approval. We don't know for sure, but I think that based on the data we've seen, there's a reasonable likelihood that they'll be approved next year at the earliest. And so we still won't know a lot about exactly how this gene therapy works. Why is there so much variability? Why is the durability being lost in the factor VIII study? And that sort of thing. And so we really are going to need to rely on post-marketing surveillance and the need of a registry to track people who have received the gene therapy. Toward that end, at the World Federation, we have initiated over these last few years the development of a gene therapy registry that will be worldwide. We'll be working with Athyn here in the United States to collect the data for all of the patients treated in the US, and then we'll be collecting the data worldwide in a number of other countries that are doing gene therapy. So this will be important to monitor over the long term to know how well it works and what any potential side effects might be from it. With that, I thank you for your attention.